everyone. I am Dr. W Somraj Singh, Associate Professor in Institute of Pharmacy at Samsung School University. So today we are going to discuss introduction to physical chemical properties, one of the primary topics in the medicine of chemistry. Understanding this concept is essential because the success of drug depends not only in the pharmacologist's activity but also on its physical chemical properties. So during this lecture, we will explore the historical development of medicinal chemistry that all the uh, physicochemical properties in drug action and important concepts such as the ionization, partition coefficient, protein binding, chelation, bioassociationism, and the stereochemistry. So let's discuss uh, with the history on the development of the medicinal chemistry as medicinal chemistry has evolved over thousands of years. Initially, medicine was based entirely on empirical observation and the ancient civilizations relied on natural products, often from plants, animals, and minerals to treat disease. In the ancient China, as the Mahong, also known as ephedra, which was used to treat the respiratory disorders. And in India, Ayurvedic natural oil serpentina for the treatment of hypertension and the mental disorders. In ancient Greek, physicians such as Hippocrates prescribed willow bark for the pain and to relieve from the fever. So although these remedies were effective, but the active constant which were responsible for the therapeutic effect were unknown. And in the 19th century, brought a revolutionary change in medicinal chemistry. So in the year 1805, Frederick Suttner asked morphine from opium. So this discovery demonstrated that a specific chemical compound could produce a defined biological effect, and in the year 1828, Frederick Williams synthesized urea in the laboratory. So this achievement is proof that the theory of vitalism and established the foundation of modern organic chemistry. In the year 1899, Bayer's Institute aspirin, one of the first successful examples of chemical modification of a natural product for therapeutic benefit. So these discoveries transformed medicine from empirical practice into a scientific discipline. And the other major contribution was that Paul Ehrlich, often regarded as the father of uh, modern medicinal chemistry, Andres proposed the receptor theory, suggesting that drug interact with specific biological target, and he introduced the concept of the magic bullet, a drug designed to selectively active that disease-causing organism while sparing healthy tissue means it does not affect the normal cell or the healthy cells. It only target to those this one like with these or microorganisms or other effective so the virus cells and all. So his work resulted in the development of the salvarsan, that the first effective treatment for syphilis and one of the earliest example of the rational drug design. So golden age of medicinal chemistry, we can see as the Alexander Fleming accidentally discovered the penicillin in the year 1928. He observed that the mold in bacterial growth leading to the development of the antibiotics. So this accidental discovery revolutionized medicine and significantly reduced the mortality from the infectious disease. And the beta-lactam ring present in the penicillin remains one of the most important structural motifs in antibacterial therapy. Today, the drug discovery follows a systematic process involving the target identification, lead discovery, lead optimizing, preclinical testing, and the clinical trials. So, medicinal chemistry play a crucial role in optimizing the molecular structures to improve potency, selective safety, and pharmacokinetic properties. Now, let us move to the physical chemical properties. So these physical properties determine how the drugs behave within the body. They influence absorption, distribution, metabolism, excretion, and the toxicity. In the short, we can abbreviate as the ADMAT. So one of the most important properties what we are going to do is about the ionization. So most of the drugs are what the weak acids or either they are the weak base. So their degree of ionization depends on the pH of the surrounding environment and the drug pKa value. So the unionized molecules are generally lipid soluble and cross the biological membrane easily. And the ionized molecules are more water soluble and tend to remain in aqueous environments such as the blood plasma. For example, aspirin is what is a weak acidic drug and is better absorbed in the acidic environment of the stomach. And as per the Anderson Hasselbalch equation help us to understand the relation between pH and the pKa. For weak acids, the equation relates the ratio of the ionized and the unionized forms of the drugs. When the concentration of the conjugate base equals concentration of the weak acid, the pH becomes equal to the pKa. So if the conjugate base concentration increases, the solution becomes more alkaline. If the weak acid concentration predominates, the solution becomes more acidic. So this equation is extremely useful in predicting the drug absorption and the distribution. Now, let's just discuss solubility and the Lipinski rule of five. So basically known as the Christopher Lipinski proposed that this rule to predict whether a compound is likely to exhibit a good oral bioavailability or not. So according to the rule of five, molecular weight should be less than 500 Daltons and the partition coefficient or the log P value should be less than 5. And the number of the hydrogen bond donors should be favored than 5 and the number of hydrogen bond acceptors should be favored than 10. So compounds satisfying this criteria generally possess favorable absorption and the permeability characteristics. Now let us discuss the partition coefficient. The partition coefficient represented as the log P measure the lipophilicity of a drug. So it is defined as the ratio of compound concentration in the octanol to its concentration in the water. A high log P value indicate increased lipid solubility and enhanced membrane permeability. However, excessive lipophilicity may reduce aqueous solubility and create formulation challenges. Conversely, very low log P values improve water solubility but may hinder membrane penetration. So therefore, medicinal chemistry strives to achieve an optimal balance between the solubility and the permeability. And another topic is also very important, that is the concept of the hydrogen bonding. So hydrogen bonding significantly influence the drug receptor interaction and the water solubility. So intermolecular hydrogen bonding occurs between different molecules and generally increase water solubility. So intramolecular hydrogen bonding occurs when within the same molecules and may reduce the water solubility while increasing lipophilicity. In here, the classical example is the comparison between the orthonitrophenol and the paranitrophenol. So orthonitrophenol forms intramolecular hydrogen bond, resulting in the lower water solubility, whereas and the low boiling point compared with the paranitrophenol. And the other topic is about the protein binding, which is a critical factor affecting the drug action. Many drugs bind reversibly to plasma protein. Albumin primarily binds acidic drugs, while the alpha-1 acid glycoprotein binds basic drugs. So only the unbound fraction of a drug is pharmacologically active. So highly bound drugs often exhibit prolonged half-lives and a slower elimination. So drug displacement interaction can also occur. For example, displacement of the warfarin plasma protein can increase the bleeding risk. So chelation complex and the important consideration in the pharmaceutical practices. And here the chelation involves the formation of a stable complex within a drug and a metal ion. That here, for example, the tetracycline antibiotic readily form a complex with the calcium, magnesium, and the aluminum ions. As a result, the tetracycline should not be administered with the milk, dairy products, or the antacid because insoluble complex reduce drug absorption. Now let us discuss the bioassociationism. So this involves the replacing the one atom of or the functional group with the another that possesses a similar physical or the chemical properties. And the objective is to improve efficacy, safety, selectivity, or the pharmacokinetic behavior while retaining biological activity. Bioassays also are classified as the classical and the non-classical. In classical bioassay follows the traditional steric and electronic principles. Examples like it includes the substitution including fluorine, chlorine, hydroxyl, amino, and the methyl groups. And in the non-classical bioassay do not necessarily obey valency rules but produce similar biological responses. An important example is the replacement of carboxylic acid group with the tetrazol ring. Uh, let's discuss about the stereochemistry. So many biological targets are chiral, therefore the spatial arrangement atoms uh, in the drug molecule directly influence its biological activity. So optical astrometers arise from chirality and release in the non-superimposable mirror images known as the N-isomers, like thalidomide. Tragedy is one of the most significant examples demonstrating the important stereochemistry. And in the geometrical isomers results for restrictive rotation around double bonds or the cyclic structures like cis and trans isomers often exhibit markedly different biological activities. A classic example is the cisplatin and the transplatin. So let us consider about some of the important discussion questions. Suppose uh, we are designing a drug intended to act with the central nervous system. Will you prioritize the high water solubility or the high log P value? Because drug must cross the blood-brain barrier. Moderate lipophilicity is generally desirable. However, accessibility possibly may comprise the aqueous solubility. Therefore, medicinal chemists must carefully balance these properties during the drug design. And before concluding, there are some of those, let's summarize some of those things like ionization and the pKa influence drug absorption. Log P determines membrane permeability. Hydrogen bonding affects solubility and receptor binding. Protein binding influence pharmacological activity and the drug half-life. Chelation can reduce the drug absorption. Bioisosteres is a powerful strategy in the drug design. Stereochemistry significantly impact efficacy and the safety. In conclusion, medicinal chemistry has evolved the traditional remedies to sophisticated rational drug design, and modern drug discovery relies on understanding and manipulating physical chemical properties to optimize therapeutic performance. By controlling the parameters such as pKa, log P value, protein binding, bioisosteres, and the stereochemistry, medicinal chemistry, chemists can design drug that maximum efficacy while minimizing the toxicity. That's what we are designed to synthesize so many derivatives by reducing the toxicity and to improve the efficacy. So, thank you for your attention. I hope this lecture has enhanced your understanding of medicinal chemistry and the physical chemical properties of drug. If you have any questions, I will be happy to discuss them. So, thank you and have a wonderful day. If you have any questions or any query, you can send to my mail. So, I'll be happy to answer that. Thank you, everyone. Have a nice day. These are some of the references which you can follow for the further readings and for further understanding. Thank you.